Okay, everybody, we're at the last part of our little section here. We're doing slides 16 through 20, or I'm sorry, we're doing slides 21 forward. So the first thing I want to talk about here is how bacteria reproduce. And one of the first things you'll notice is it says asexual. So that means without regards to sex. So with bacteria, it's not like there's a male and a female version of bacteria reproducing with one another. Asexual means without sex. Okay? Now, the process in which bacteria divide has its own special name that's different than mitosis. It's called binary fission. Now, ultimately, it produces the same thing that mitosis produces, which are genetically identical daughter cells to the parents. But it doesn't have to go through all the steps like prophase, metaphase, anaphase, and telophase. It doesn't have to build a spindle apparatus or any of those special bells and whistles that we saw with mitosis. It's a much simpler process. Remember that with bacteria, DNA is not found in numbers of chromosomes that are separate from each other in X-like shapes. Instead, the DNA in bacteria is found in a simple ring, and that ring is found in the nucleoid region of the cell. There's no special nuclear membrane where everything is held. It's just kind of floating freely in the cytoplasm, a ring of DNA in the nucleoid region of a bacterium. Okay? Now, what has to happen is ultimately the same as what happened in mitosis. You need to double up the genetic content in order to have two daughters that have a full set. If you just split it down the middle and cut the ring in half, then each daughter would only receive half the amount of DNA they need, half of an instruction set, and they need a full set for that to live on. Okay, so we got to copy that single ring-like chromosome so each daughter can have one. And of course, the bacteria itself has to be large enough to split into two, so we want the cytoplasmic volume to also be increased during this process. Okay, so let's look at it in terms of 5 steps. Number 1, attachment of the chromosome to a special plasma membrane site indicates this bacterium is about to divide. So again, very simple small cell here. Here is the ring of DNA. And here you can see it has touched a special receptor in the cell membrane, which is kind of the kickoff point to dividing into 2. Number 2, the cell is preparing for binary fission.
We are meeting in the cafeteria.
By enlarging its cell wall, plasma membrane, and overall volume. So you see it's getting longer here lengthwise. And you can also see that that second DNA that's being copied is currently happening, and it's that purple one behind it. Number 3, you can it says DNA replications produce 2 identical chromosomes. Cell wall and plasma membrane begin to grow inward. So now you can kind of see they're drifting apart to either ends. And you now start to see that there's kind of a pinching inward of the sides as this cell is actively starting to divide into 2 cells. Number 4, as the cell elongates, the chromosomes are pulled apart. Now they're completely separate. Cytoplasm is being divided evenly. And then in number 5, a new cell wall and plasma membrane has divided one cell into 2 identical daughter cells. This DNA is identical to what they started with up there. Okay, and that's the process of binary fission for bacteria. Okay, the last thing that we were going to talk about that matches our learning targets is about cancer. So we said in class that cancer was uncontrolled, and you can write this above if you'd like, uncontrolled cell division. So normally there are rules about cell division, and they start and stop according to a program which all cells need to follow. But what happens when they don't is we have uncontrolled cell division. We already said in a prior lecture that when there's a lot of cells around each other and they're touching, they get a signal to stop dividing. They're crowded enough, they don't want more. We also said that if you're an animal cell, you want a solid layer beneath you, and you don't want to be on top of a pile of cells. If you evade those 2 rules, those 2 expectations, you're probably a cancer cell, okay? A cell that has uncontrolled cell division. Now, there's 2 big things that are kind of the main reasons why cancer happens. The first one has to do with something called proto-oncogenes. So proto-oncogenes are normal genes that promote the cell cycle and they prevent the cell from, abnormally dying early or blowing itself up or anything like that. Okay, so it provides some normalcy, a moving ahead in the cell cycle. But if a mutation happens to one of these proto-oncogenes, what can happen is it turns it on, and it almost like accelerates the process. So if we use a car analogy, if you slam on the gas, you start moving forward very, very quickly. Same thing with a cell cycle. If there's a mutation that causes something to go wrong with the proto-oncogene, which is normally pushing cell cycle forward in a normal controlled way, it suddenly speeds forward out of control and produces all kinds of products that shouldn't be being produced at that speed. The second big reason behind why cancer can develop from genes are mutations in what's called tumor suppressor genes. Okay, so a lot of us know what tumors are. They're masses of abnormal cells. Suppressors are things that slow down or stop. Okay, so a tumor suppressor gene is a gene that slows down or stops tumors or abnormal cells from continuing on. Okay, so they're actually a good thing. But when there's a mutation or when these genes malfunction, you take away that ability to stop or slow down abnormal cells. So back to the car example, it would be a car where the brake pedal doesn't work. You're trying to slam on the brake pedal and it doesn't work. The cells keep on going, they keep on dividing, okay? So over half of all cancers involve one of these faulty, it's a very special one called P53, tumor suppression genes. It takes away its brake pedal and the cell cycle just kind of moves forward quickly out of control. Now, as far as cancer itself, a lot of questions, how do you get it? Well, there's two real common answers, genetically and environmental. Okay, so cancer can run in a family. Environmental. There we go. Okay, for example, breast cancer. Breast cancer can be present in a lot of members of certain families. And it's nothing they did wrong, it just happens to be something that's passed on from generation to generation. So if you have a gene or you have a record of cancer in a high number of individuals in your family, that may lead to questions, about your own future and whether you want to take steps ahead of time to proactively perhaps mitigate the fact that cancer could be coming your way. There are things you can do. Environmentally, when we're exposed to things that we shouldn't normally be exposed to, like radiation and cigarette smoke and too much UV light, all of those things also have the ability to mutate genes, like your proto-oncogenes and your tumor suppressor genes. So those could be the start of cancer. And it always starts with just one cell. So one cell will ultimately become mutated and become a cancer cell. That one cell will divide into two into four into 8, 16, 32, 64, 128, 256, 512, 1024, 2048. I'm already in the 1,000. It took me about 12 seconds to get there. So you can see that it happens quick and they build up fast. So it's very important that we get a control of them, diagnosed, and then treated as fast as possible. When a single cell gets converted to a cancerous cell, we call that process transformation. That's the initial event that occurs, and then that doubling up happens as that cell starts to divide. Those cancer cells will eventually form a tumor, which is a mass of abnormal cells. We've mentioned that a little bit earlier. And that tumor can be one of two types. It can be what we call benign, where the abnormal cells remain at the original site. They're kind of halted there. They are abnormal, but they're not really doing a whole lot. And that's actually the preferable diagnosis we want, is that they're benign. Now, if they're not benign and they're still dividing and they're on the move, we call them malignant tumors. They're starting to invade the surrounding tissues, and then they go through what's called metastasis, where ultimately one or two cells or even more of them may break off, find a way into the bloodstream, and then get redeposited somewhere else in the body and start to grow there as a secondary tumor, a secondary site. Okay. Another bad thing that cancer cells do is they exhibit something called angiogenesis. And angiogenesis, angio refers to blood vessels, genesis is something new. So they actually draw new blood vessels to themselves to create their own blood supply. And of course, that's a bad thing. You don't want cancer cells having all the nutrients they want. We want to starve them out of nutrients. So essentially, they cease to exist. But that's what they do when they go too far, and that's ultimately not a good thing. This here is kind of a timeline showing how cancer can develop in breast tissue. So initially we have that transformation event where one cell goes cancerous, and then it starts growing from there, from that single cancer cell. Now it's in tumor form. Maybe this is grown and divided a number of times here. And as it progresses, you can see that tumor mass is now probably double or triple the size it was here as the cells start to invade the neighboring tissue. As it progresses, it will continue to grow in size, but now we start to see the metastasis, the breaking off of those cancer cells, winding up in blood vessels, which allow them to travel to other parts of the body and then set up and start growing there as well. So if it started in this woman's breast right here and it took a ride to a new location somewhere in her abdomen here, now we have a secondary cancer site and the cancer is spreading. Okay, so she's going to want obviously to get treatment on that. And as far as treatments go, we have chemotherapeutics, so chemotherapy, where we infuse chemicals in their body that are known to inhibit cancer cells from dividing. We have radiation treatments, targeted high-energy laser treatments, treatments of radiation that are directed specifically at those cancer cells. And we're even getting into what we call personalized medicine, where we take the cells that are working in your body fighting the cancer, we enhance them in a lab and put them back in the patients so they have the best of what their body can fight the cancer with. A lot of research right now is also being focused on something called a caspase. Caspases are sets of proteins that bring about apoptosis in cells. Apoptosis, we said earlier, was programmed cell death. So ultimately, if we can have apoptosis just happen in the cancer cells, that would be a great thing, where the normal healthy cells would then be spared. Because in like chemotherapy and radiation, other cells are affected, normal healthy cells, by those same treatments, and they that usually has negative outcomes for the patients. They lose their hair, for example. They feel nauseous and sick all the time because those cells are also affected by the treatment they're getting. But some cells do have to continue to divide throughout their lifetime, and they're not cancerous. Like your blood cells are constantly being replenished as they wear out. We form new ones. Your skin cells, the same thing. We lose skin cells off the top of the surface of all parts of our body, and they grow back from the base layer up. We have digestive system cells that we lose as food passes through our digestive tracts, and they're replaced, as well as respiratory cells. We breathe in dust and debris, and we lose cells in our respiratory system, and they're replaced all the time throughout our life. Where a lot of the other cells are still kind of arrested in that GO stage unless we're injured. There's not really a need for them to actively divide as much. Okay? All right. So our last slide here, we'll just kind of go through a little table here. Differentiation is when cells have different jobs, like a liver cell, a brain cell, an eyeball cell. Normally, that happens, but in cancerous cells, they don't really have those specific roles anymore. Now they're just kind of classified as cancer cell. It's different than all your other cells. Normal nuclei, nope, there's something different. There's a mutation inside of the nucleus that's causing it to be cancerous. They do not undergo apoptosis. We wish they would, because we don't want them. They contact inhibition when they're surrounded by other cells. Do they blow themselves up? No, they keep growing, they keep dividing, where normally cells would arrest cell division at that point. Organization, they lack organization, where normal cells have organized cell interiors. We said that they metastasize. They can also have angiogenesis affect them, whereas normal cells do not. Okay? All right, so this is the completion of our 8.2 focus of cell cycle, mitosis, and cancer. This is the end of the notes. So the next thing that we're going to do is we're going to have our assessment coming up. So keep checking Schoology for assignments if you are watching this because of an absence. And I'll see you in class. Okay, bye-bye.